convener of this conference, Dr. Sasikant Dwivedi, and the organizing secretary of this conference, Dr. D.K. Singh and Dr. Saha. Here you see the title of the symposium is on laser in biology, medical science, atmospheric science, and climate change. That means here you see laser is generally based on spectroscopy. Therefore, here you see that spectroscopy is the branch of physics which has one potential application in almost every branch of science. Here you can see biology, medical science, chemical science, physical science, agriculture science, environmental science, so on and so forth. And the common spectroscopic technique which are being used by the scientists working in almost every branch of the sciences are UV spectroscopy, fluorescence spectroscopy, Raman spectroscopy, infrared spectroscopy, so on and so forth. Here in the present part, I am going to talk about the LIPS. LIPS means laser-induced breakdown spectroscopy. But before coming on the topic of my talk, whenever I go to address such type of audience, then I usually send a message. I usually try to convince the audience that the knowledge of any branch of science is incomplete without the knowledge of physics. Why I am saying like this? Because if you want to explain anything, any phenomena occurring in our daily life, then you have to take the help of physical principle of physics. I am giving one simple example. For example, you are sitting in a drawing room with your friend, and in the drawing room you have plenty of light, that you have light from LED, tube light, bulb, and so on and so forth, and you are just tapping your friend very comfortably. Imagine when the light suddenly gone back. What will happen? You cannot see your friend or thing just beside you. But when the time pass away, then slowly and slowly you can see your friend or thing which are just beside you. Can you imagine how this will happen? This can again explain with the help of the physical principle. Here you see you can equate the function of our eye with the function of camera, photographic camera. In the camera there are three main things. One is the shutter, one is the lens, and the third one is the retina. Here you see in the camera, the shutter controls the light entering on the lens of the camera and focus on the retina. So if you have an ample amount of light, then the opening of the shutter of the camera is very, very small, so that light entering on the photographic plate is not so intense, otherwise your photographic film becomes black. Similarly, in your eye, we have a iris, and this iris acts as a shutter in the photographic camera. We have a lens also in our eye, just like camera, and we have a retina, just like photographic plate. So when you are sitting in the drawing room, having ample amount of light, then what will happen? In that case, the opening of the iris is very, very small, so that not much light from your drawing room entering the eye and focus on the retina, otherwise your retina will be damaged. So in this way, the opening of the iris is very, very small, and you can see your friend or thing very comfortably. But when the light went off suddenly, what will happen? You cannot see because the opening of the iris is very, very small. So the ample amount of light, sufficient amount of light is not entering on your eye, and it is not being focused on the retina. Therefore, you cannot see the thing just beside you. But when the time passes away, what will happen? The opening of our iris is going to increase slowly and slowly. Then when the opening of the iris is very large, then even the small amount of light is entering on your eye, and you are comfortably see your friend or thing just beside you. So this is in this way you can just imagine what will happen when you are sitting in the dark drawing room and the light is not sufficient. There is no light. And what will happen when the light suddenly came back? What will happen? You cannot see. Your eye is just क्लोज ओपन क्लोज, जिसको कहते हैं चकाचौंध हो जाता है, चकाचौंध हो गया। क्यों ऐसा हुआ? क्योंकि एट दैट टाइम इन द डार्क रूम द ओपनिंग ऑफ द iris is large, is very large, and when the light came back suddenly we have a ample amount of light and your opening of iris is very large. So if the opening is very large, then the large amount of the light can enter in your eye and it can damage your retina. So in that case, you are not comfortable and just light चकाचौंध हो जाता है और आपकी आंख खटने बंद होने लगती है, खुलने लगती है, बंद होने लगती है. So that retina cannot be damaged. When the time pass away again, the opening of the iris goes on decreases, and so that not all light from the time will enter your eye and you can see the things very comfortable. हम लोगों ने ये भी सुना है कि हमारे ग्रैंड मदर या ग्रैंड फादर जब एज ज्यादा हो जाता है तो उनको दिखाई नहीं देता है, दूंगा फिर दिखने लगेगा. क्या हो जाता है जब एज जैसे जैसे बढ़ने लगती है? तो जैसा मैं बताया कि हमारे आंख में लेंस है, लेंस के ऊपर एक लिक्विड परत जमने लगती है. जब लेंस के ऊपर कोई परत जम जाएगी तो उसका ट्रांसमिशन पावर बहुत कम हो जाएगा. जब कम हो जाएगा तो आपको दिखाई नहीं देगा. जब दिखाई नहीं देगा तो आपके पास डॉक्टर के पास जाना पड़ेगा. तो डॉक्टर क्या करता है? हाथ टूट गया, अब कहते हैं एक्सरे करा लीजिए. एक्सरे में होता क्या है? एक्सरे का जो वेवलेंथ होता है वो यूवी विजिबल से कम और गामा रे से ज्यादा होता है और उसकी पेनिट्रेटिंग पावर बहुत ज्यादा होती है. जैसा हम जानते हैं कि हमारे बॉडी जो है 80% लिक्विड से बना हुआ है, लिक्विड वाटर. तो उसका एनर्जी इतना ज्यादा होती है एक्सरे का कि वो आपको लिक्विड मटेरियल को पेनिट्रेट कर जाता है, लेकिन जो आपके पास बोन है वो सॉलिड है उसको पेनिट्रेट नहीं कर पाता है. तो उसका आपको पिक्चर दिखाई देने लगता है. और जहां आपका बोन टूटा हुआ है वहां तो एयर है और वहां से एयर पास हो जाती है, तो आपको एयर फ्रैक्चर जो है बोन में जो फ्रैक्चर है वह बहुत आसानी से दिखाई देता है. इस तरह से मैं बहुत सारी एग्जाम्पल दे सकता हूँ, उसमें नहीं जाऊंगा. हियर यू सी स्पेक्ट्रोस्कोपी. LIBS means laser induced breakdown spectroscopy. So first of all, I will tell about little bit the पढ़ाएंगे. Here you see, spectroscopy deals with atoms and molecules and covering physics as well as chemistry. But whether it is a branch of physics or chemistry, it has potential application in the problem relating to nanotechnology, biological science, medical science, environmental science, and agricultural science, and so forth. As I said that the common spectroscopic technique, which are the sciences may be categorized as Raman spectroscopy, infrared spectroscopy, UV visible spectroscopy, fluorescence spectroscopy, FTIR spectroscopy, so and so forth. In the present talk, I am going to describe a new laser-based technique, and the name of that technique is LIBS, laser-induced background spectroscopy. Here you see, laser is the full form of this LIBS is laser-induced background spectroscopy, and it is which has wide application starting from the alphabet A, which stands for archaeology, to the alphabet Z, which stands for zoology. You can just imagine the importance of this technique. This is the elemental composition of the target on another planet like Mars to study its geological layer, because NASA Curiosity rover, which landed on Mars in the first week of August 2012, carries a LIBS equipment for exploring the geological layer of red planet. Here you see, this is the photograph of the cover, and here you see the green one. Green one is the laser light, and the laser light is being focused on the rock surface on the Mars, and the white one here is the white one is the laser induced plasma, and light in this plasma contains all these spectral signatures of the constituent which are present in this rock. Therefore, if you collect the light from this plasma and if you analyze it, then you can get the information about the constituent, about the element in this rock without touching the rock, without approaching the rock. You can get all the information. This technique is not only gives the information about the surface of the rock, but if you focus the laser light at the same point for a longer period of time, then you can get the information about the first layer, second layer, third layer, fourth layer, and so on and so forth. Here you see this is the actual photograph. Actual photograph of the laser beam focused at the surface, and here you see you can see the laser pit very clearly, and in this way you can get the information about the elemental constitution of the different layer in this present system. By seeing the importance of this technique, by by seeing the importance of this technique, India has also included the list equipment in Chandrayaan-2 mission is not destroyed. A microgram sample is only evaluated and the sample is intact. So you can say this technique is not destructive. You can reuse the sample without after the analysis. Point detection capability. For example, if you want to see what are the elemental constituents at the black part, blue part, and green part, you can do it very easily by just by focusing the laser at the black part, then green part, then blue part. So in this way, you can get the spatial information, the variation of the element, especially distributed on any surface. You can do it very easily without removing this, the material from the surface. So this technique is useful for simultaneous analysis of all the elements present in the periodic table. No other technique can tell the information about simultaneous analysis of all the elements present in the periodic table. This technique is useful for quantitative measurement of minor, which is present as a trace level, as well as measure main constituents of the matrix. This technique has a remote analysis capability. For example, you have a radioactive substance and you do not want to touch the substance. So, and if you want to find out what amount of radioactive elements are present in that sample, you can do it very easily because we are not going to touch this sample here. We can just focus the laser light and you can get the information. So this technique is useful for remote analysis capability. For example, you have a big furnace, aluminum alloy furnace, and if you want to monitor the elemental present in this aluminum alloy without collecting the material from the furnace, you can do it very easily by focusing the laser light in the furnace. You can get all the information. If any material, the concentration of the material is less or high, then accordingly you can take the decision at the spot. So that way, this technique has a capability for in situ, online, and real-time analysis. So by seeing the importance of this technique, we have developed the experimental facility of LEAPS in the Department of Physics, University of Allahabad, and it started multidisciplinary research like topic here. Our group is involved in the study of a variety of materials like geological materials, archaeological materials, biomaterials, energetic materials, plant materials, and food materials. We are also involved in the monitoring of toxics and heavy elements present in the different industrial waste, soil, water, plant, and plant product. We have published a large number of papers. If somebody want to take that particular paper in that particular materials, I can give you very easily. But in the present talk, a brief discussion of the work related to the study of gallbladder stone will be discussed. Here you see, gallstone formation in the gallbladder is one of the leading disease throughout the world, and it is one of the most painful disease. And if the stone remains in this gallbladder here for a longer period, then it can trigger many complications, just like cancer. आपने gallbladder cancer सुना हुआ क्यों होता है? बहुत दिन तक जब स्टोन gallbladder में पड़ा रहता है तो रगड़ से जो है gallbladder में कैंसर उत्पन्न हो जाता है. तो इसलिए इसका एनालिसिस करना, कैसे ये इसका न्यूक्लिएशन होता है, कैसे ये स्टोन कैसे बढ़ता है, इसकी बहुत जरूरी है स्टडी करना. आगे चलिए. Here you see, to know the mechanism formation of the individual gallstone, it is important to study various groups of gallstone. And as you know that the gallstone is basically classified in 3 categories: cholesterol stones, pigment stone, and mixed stone. And as you know that the stone may occur both in male and female as well as in the child. Complete pathogenesis is still unclear. It may be due to aspect of dietary intake, including the quality of the drinking water. It may be due to inherent metabolic disorder of the patient. The complete knowledge of chemical constituents of gallstone are not fully identified. Therefore, our aim is to achieve this goal. We have used LIFT technique to perform the elemental analysis of different types of materials. Next. Here you see, our main aim is just to study the role of different mineral element, particularly magnesium, in the formation of the gallstone. What is the role of magnesium in the formation and growth of the gallstone? Here you see, for such type of study, we have selected 4 types of gallstone. here you see this gallstone is big, nearly 5 centimeter area. Here you see this gallstone, we have a small granule. That means small stones, and these small stones joined together by some material and form a big gallstone. But if you see this gallstone, then this is not made up of a small granule, small stone. This is a very big gallstone, and size of these two gallstones are more or less similar, equal. And if here you see, this is the another type of gallstone, and all these gallstones are obtained from a सर्जरी ऑफ ए सिंगल पेशेंट। ये सब गॉलस्टोन एक ही गॉल ब्लैडर में थे, एक ही पेशेंट के गॉल ब्लैडर में थे। और ये दूसरा तरह का गॉल ब्लैडर स्टोन है, जो कि ये गॉल ब्लैडर एक दूसरे पेशेंट के गॉल ब्लैडर में एक ही स्टोन प्रेजेंट था, और एक पेशेंट के गॉल ब्लैडर में इतने सारे स्टोन प्रेजेंट थे। एक पेशेंट के गॉल ब्लैडर में ऐसा स्टोन था जो कि बहुत बड़ा तो था, लेकिन जो उसका जो ग्रेनुल है वो छोटे छोटे साइज से बना हुआ था। इस तरह से गॉल स्टोन थे, छोटे छोटे थे, लेकिन इसमें क्या है कि वो ज्वाइंट कर गए, एक दूसरे से कंबाइन कर गए हैं, और इसमें कंबाइन नहीं किए हैं, और ये बहुत बड़ा गॉल स्टोन है। अगर हमारा ऐसा मानना है कि इस गॉल स्टोन को बहुत छोड़ दिया गया होता तो ये भी करीब इतने बड़ा हो गया होता। तो हमारा रोल ये है, हम ये देखना चाहते हैं कि कौन सा एलिमेंट है जिसके चलते हम इस तरह का गॉल स्टोन मिलता है, इस तरह का गॉल स्टोन मिलता है, इस तरह का गॉल स्टोन मिलता है, और इस तरह का गॉल स्टोन मिलता है। इसके लिए हमने क्या किया कि इसके सरफेस पर लेजर को फोकस किया और उसका एलिमेंटल कॉन्स्टिट्यूएंट पता किया। इसके बाद क्या किया कि इस गॉल स्टोन को दो भाग में काट दिया, उसके सेंटर पर फोकस किया, या कह सकते हैं कि पहले पेरीफेरी से शुरू किया, पेरीफेरी पे गया 1 2 3 4 सेंटर गया, फिर 1 2 3 4 पेरीफेरी गया, और देखा कि किस तरह से एलिमेंटल का वेरिएशन हो रहा है, कंसंट्रेशन का वेरिएशन हो रहा है। जैसा कि अभी मैं कहा कि ये टेक्निक जो है पॉइंट डिटेक्शन कैपेबिलिटी है, और इस टेक्निक में हम देख सकते हैं कि कैसे एलिमेंट स्पेस वाइज वेरी कर रहा है। उस एडवांटेज का फायदा उठाते हुए हम लोगों ने इसकी एनालिसिस किया। आगे चलिए। हियर यू सी दिस इज द लीफ स्पेक्ट्रा रिकॉर्डेड एट द सरफेस of the gallstone and as well as at the center of the gallstone. Here you see the red one is the leaf spectra recorded at the center, माने दो भाग में कर दिया, उसका सेंटर पर जब फोकस किया तो रेड वन आया, और पेरीफेरी पर देखा तो ये ब्लू वन वाला है। ये आप देख सकते हैं कि इंटेंसिटी जो है मैग्नीशियम लाइन की इंटेंसिटी जो है सेंटर पर ज्यादा है और पेरीफेरी पर कम है। इसका मतलब कि मैग्नीशियम का कंसंट्रेशन सेंटर पर ज्यादा है और पेरीफेरी पर कम है। आगे बढ़िए, आगे बढ़िए। तो हमने यही है, उसके पीछे नहीं है, वही है। हाँ देखिए, फिर ये भी है, यहाँ पर भी देख सकते हैं कि पोटेशियम या कैल्शियम का भी कंसंट्रेशन जो है सेंटर पर ज्यादा है और पेरीफेरी पर कम है। आगे बढ़िए। हमने क्या किया कि एक पेरीफेरी से लेकर के दूसरे पेरीफेरी तक डिफरेंट पॉइंट पर लेजर को फोकस किया और लीफ स्पेक्ट्रा रिकॉर्ड किया, और एक जो स्पेक्ट्रल लाइन है उसकी इंटेंसिटी को मेजर किया, एरिया अंडर द कर्व मेजर किया। तो हमने देखा कि अब जब ये कैल्शियम की बात करें, हम ज्यादातर इस पर फोकस करेंगे मैग्नीशियम पर। जब देखेंगे कि कैल्शियम जो है एक पेरीफेरी से शुरू करते हैं तो वहां कम रहता है, फिर बढ़ता है और सेंटर पर जाकर मैक्सिमम हो जाता है, और फिर जब सेंटर से दूसरे पेरीफेरी पर बढ़ते हैं तो वहां पर भी इंटेंसिटी कम होने लगती है। आगे चलिए। अब देखिए, अब वो तो जो है बिग स्टोन था, इंटेक्ट बिग स्टोन था। अब दूसरे स्टोन की बात करने जा रहे हैं जो कि छोटे छोटे स्टोन आपस में कंबाइन करके बड़ा स्टोन बना दिया। अब उसमें देखिए, दिस इज द माइक्रोस्कोप इमेज ऑफ द ग्रेन्यूल्स ऑफ दिस gallstone, and here what we expect is magnesium is one of the most abundant mineral in the cell with an essential role in fundamental biological function। और मैग्नीशियम का ही कंसंट्रेशन ऐसा वेरी कर रहा है कि स्टोन या तो छोटा छोटा रह जा रहा है या बहुत बड़ा हो जा रहा है। और इसको जो है च्यूंग एट अल ने एक डोज रिस्पांस रिलेशनशिप से स्टडी किया, और ये पाया कि ये रिपोर्ट किया कि हायर कंजम्पशन ऑफ मैग्नीशियम इज एसोसिएटेड विद रिड्यूस रिस्क ऑफ गॉलस्टोन डिजीज। जब मैग्नीशियम का कंसंट्रेशन ज्यादा हो जाता है तो स्टोन जो है छोटा होने लगता है या उसके चांस कम हो जाते हैं। इस चीज को हम लोगों ने देखा। अब देखिए, अब आपको पहला याद होगा कि जब हम लोगों ने बड़ा इंटेक्ट गॉलस्टोन लिया था तो मैग्नीशियम का कंसंट्रेशन सेंटर पर बहुत ज्यादा था। अब यहाँ पर देखिए, ये देखिए रेड वाला जो है आपका सेंटर पर है और ये ग्रीन वाला जो है आपका पेरीफेरी पर है। यहाँ देख रहे हैं कि पेरीफेरी पर मैग्नीशियम का इंटेंसिटी ज्यादा हो जा रही है, मैंने मैग्नीशियम का कंसंट्रेशन पेरीफेरी पर ज्यादा हो रहा है इन कंपेरिजन टू दी सेंटर। इस तरह से ये है। आगे बढ़िए। इस तरह से आपने देखेंगे कि क्या नाम है, ये पोटेशियम का भी कंसंट्रेशन पेरीफेरी पर ज्यादा हो रहा है। और आगे बढ़िए। अब इसमें क्या किया कि हमने मेजर किया, इसमें तो अभी हमने दिखाया इस वाले स्टोन में कि जब पेरीफेरी पर मैग्नीशियम कंसंट्रेशन कम रहता है और जब सेंटर पर जाते हैं तो बढ़ता है, जब फिर दूसरे पेरीफेरी पर जाते हैं तो फिर कम हो जाता है। अब ये देखिए इसके केस में, इसमें क्या हुआ कि पेरीफेरी पर मैग्नीशियम की इंटेंसिटी ज्यादा है और सेंटर पर कम हो गया, फिर दूसरे पेरीफेरी पर जाते हैं तो बढ़ गया। इसका मतलब क्या है कि छोटे वाले में मैग्नीशियम का कंसंट्रेशन सरफेस पर ज्यादा है। इसको ये भी कह सकते हैं कि जब स्टोन बनना शुरू हुआ होगा तो जो पेशेंट है वो ऐसा डाइट ले रहा था कि मैग्नीशियम ज्यादा था, मैग्नीशियम ज्यादा, उसका डिपोजिशन मैग्नीशियम का सरफेस पर ज्यादा होगा। अभी हमने देखा कि चाइनीज ने एक रिसर्च किया था जो डोज रिस्पांस रिलेशनशिप से, और पाया था कि अगर मैग्नीशियम का कंसंट्रेशन कंजम्पशन बढ़ेगा तो उसका जो गॉल स्टोन का फॉर्मेशन का डिजीज जो है कम हो जाएंगे चांसेस। इसीलिए इस केस में ये स्टोन बड़ा नहीं हुआ, इसके तरफ बड़ा नहीं हुआ, लेकिन छोटा छोटा कई स्टोन बन गए और आपस में जुड़ करके जो है बड़ा स्टोन बना दिया। आगे चलिए। इसी तरह से एक ग्रेनुल के था, ये दूसरा ग्रेनुल में उसी तरह का हमको रिपोर्ट मिला है, उसमें भी उसी तरह से जो है सेंटर कम है और पेरीफेरी पर ज्यादा। इस तरह से हम लोग कई ग्रेनुल, उसमें देखे ना इसमें कई ग्रेनुल्स हैं, हम लोगों ने कई ग्रेनुल्स का स्टडी किया और सब में करीब करीब इसी तरह का वेरिएशन मिला। तो क्या ये दिस इज द सेंटर पार्ट ऑफ द ग्रेनुल 2 है। ग्रेनुल्स इज डोमिनेटेड विद लो मैग्नीशियम whereas periphery is dominated with high magnesium and low calcium. This is the result of opposite to this big intake gall stone. आगे चलिए। Now अब कंक्लूजन में क्या कह सकते हैं, अगर यदि आपसे कोई क्वेश्चन करे कि आप देख रहे हैं कि ग्रोथ ऑफ गॉल स्टोन बेस्ड ऑन मैग्नीशियम, ये कोई क्वेश्चन करे इज मैग्नीशियम इंटेंसिटी इज हायर एट पेरीफेरी एज कंपेयर टू सेंटर पार्ट ऑफ द स्टोन, एंड इफ द आंसर इज यस पेरीफेरी पर मैग्नीशियम कंसंट्रेशन ज्यादा है, तो उस केस में क्या होगा, ग्रोथ ऑफ द स्टोन स्टॉप्स एंड यू विल गेट ए नॉन ग्रेनुलर स्टोन। एंड इफ द आंसर इज नो, मैग्नीशियम कंसंट्रेशन वहां पर पेरीफेरी पर कम है, तो आपको मिलेगा बड़ा स्टोन मिलेगा। Thus the formation mechanism of granular gallstone is different from non-granular gallstone obtained from the patient from the same region। एक ही रीजन के, जैसे माने गाजीपुर या बनारस का दो पेशेंट का लिया जाए, दोनों पेशेंट एक ही रीजन के थे, लेकिन इस तरह का वेरिएशन मिला था। पीछे जाइए, इतना तेज मत बढ़िए, यही आ जाइए, आइए आगे आइए आगे आइए। हाँ, here you see in conclusion what you can say that magnesium is one of the most abundant mineral in cell with an essential role in fundamental biological function. Granule stone growth depends on the consumption of magnesium, that is the growth of the stone stops if the magnesium content dominate on the periphery, and the similar result is also obtained by the another scientist, Chinese scientist, by the dose response relationship, and they reported that higher consumption of magnesium is associated with a reduced risk of gallstone disease. This paper is already published in Atoms. आगे बढ़िए। Okay, now ठीक है, एनालिसिस ऑफ द मल्टीपल, धीरे धीरे बढ़ाइए, बहुत तेज बढ़ा रहे हैं। एनालिसिस ऑफ द मल्टीपल गॉल स्टोन obtained from the single patient to know the process of nucleation. Here you see, this is the photograph of a large number, nearly 80 stones we observed from a single patient, and we want to see what is the process of nucleation or growth of in this such type of gallstone, and we assume that this is more or less similar here, because in that case also we get a small granule, but these granules are joined together and form a big granule. आगे बढ़िए तो। So here you see, this is the leaf spectra. I have shown you 80 gallstones. We have recorded many gallstones, and here you see, this is the leaf spectra of 4 gallstones. Small gallstone. This is the gallstone A, gallstone B, gallstone C, gallstone D. And here you see if the feature of the spectral line similar in all gallstones, and then intensity is also similar in all, then you can say that the mechanism of the formation of the every gallstones are similar in nature. Okay, आगे बढ़िए. So this is the what I said, that if you regard the plasma after some longer time, then you get the information about the type of molecule. So this is the spectra of the C2 band, and this C2 band is absorbed due to the presence of cholesterol and bilirubin in the gallstone. This is the other spectra of the type of the CN molecule. Again, this is present due to the presence of the organic compound. This is the another spectra of the diatomic molecule. So we have measured the intensity of the spectral line of different elements, for example, magnesium, calcium, potassium, and sodium. And here you see that if you go from periphery to center, the intensity of magnesium line goes down, decreases and become minimum at the center. Similar in the case for calcium also, and similar in the case for potassium as well as for sodium. And this result is also same as if you analyze the another gallstone. We have an active one. We have analyzed many gallstones, but here I am showing the result of two gallstones. And similar figure is observed in the gallstone B. So, what you can conclude that if you have a small stone, then the intensity of the magnesium and periphery is large, less in middle, very less in center. That means the concentration of magnesium is decreasing also. And what the Chinese scientists said, if you take the heavy amount of magnesium, then the growth of the stone stops. And similarly, here we are observing the same thing. So this is the information about the organic elements. These organic elements are absorbed with the decomposition of the organic compound like cholesterol, bilirubin, and so forth. And here we are not getting a similar trend. That means larger बीच में कम हो गया फिर बढ़ गया, larger बीच में कम हुआ बढ़ गया. तो एक सिस्टमेटिक ट्रेंड नहीं मिल रहा है. इसका मतलब ये होता है कि ये जो ऑर्गेनिक कंपाउंड है इसके ग्रोथ में कंट्रीब्यूट नहीं कर रहा है. ग्रोथ से जो कंट्रीब्यूट कर रहा है वो मिनरल एलिमेंट कर रहा है. आगे बढ़िए. इस तरह से हम लोगों ने बैंक को भी मेजर किया. इसमें भी कुछ एक सिस्टमेटिक ट्रेंड नहीं मिल रहा है. तो फिर कह सकते हैं कि ये बैंड जो आ रहे हैं ऑर्गेनिक के कंपाउंड के चलते आ रहे हैं और ऑर्गेनिक कंपाउंड ग्रोथ को कंट्रोल नहीं करते हैं. आगे बढ़िए. If you see here the aluminium, copper and iron are following the same trend. But if you see the aluminium is heavy heavy metal, and this is not controlling the growth of the stone. आगे बढ़िए. So we have measured the intensity ratio of the ionic versus neutral line, and we are getting the same trend. And by seeing this one, we can say that this one is more harder than less like this. आगे बढ़िए. In the case of the calcium is absorbed due to the presence of calcium carbonate, and we are getting the same trend here. And if the calcium carbonate is large at the periphery, that means the periphery part is more harder, and if it is less harder here and less harder here. Similarly, also we can calculate the hardness of the sample by this relation also. आगे बढ़िए. Now I am coming in the result of a single stone. One single stone obtained from a single patient in a gallbladder. Here you see, this is the least spectra. If you remember here, this is the intensity of spectral lines of magnesium. Magnesium here, if you see here, if you remember in the case of the small gallstone, the intensity of magnesium at the periphery was high. But if you see here, it is small and here it is high. At the center it is high. आगे बढ़ी करते. तो this is the magnesium. आगे बढ़ी करता. और ये है पीछे जाइए. ये है इतना. तो आप ये जो है अपोजिट ट्रेंड मिलता है इस केस में. इसमें जो है पेरिफेरी पर ज्यादा रहता है और सेंटर पर कम रहता है, लेकिन इसमें जो है इसका जस्ट अपोजिट हो जाता है. ये हमने मेजर किया पेरिफेरी पर और सेंटर पर देखा कि इंटेंसिटी ज्यादा है, पेरिफेरी पर कम है. यहाँ पर दूसरी तरफ देखा एक सिमिलर ट्रेंड ऑब्जर्व हो रहा है. ये ट्रेंड अपोजिट है जिसमें हमको 80 गैलेक्टोन मिले थे. और आगे ये फिर दूसरा हार्डनेस कैलकुलेट किया गया. आगे बढ़िए. अब फिर देखा हमने कि ये एक स्ट्रेन गैलेक्टोन के लिए है और ये सिंगल वाले के लिए है. सिंगल वाले में कैल्शियम जो है पेरिफेरी पर ज्यादा है, इसमें लो है. यहाँ ज्यादा है, लो है. तो मैंने अपोजिट है. दोनों का मैकेनिज्म जो है, जहाँ पर मल्टीपल स्टोन है उसका मैकेनिज्म अलग है और जहाँ पर सिंगल गैलेक्टोन मिल रहे हैं उसका मैकेनिज्म अलग है. चलिए आगे बढ़िए. अब इसको हमने चारों स्टोन का यहाँ पर समराइज कर दिया. आप इसमें देखेंगे तो सेंटर से इंटेंसिटी बढ़ना शुरू हो रही है, पेरिफेरी पर मैक्सिमम हो जा रही है. और यहाँ पर देखेंगे इस वाले में भी, इसमें भी क्या है, सेंटर से बढ़ना शुरू है और पेरिफेरी पर बढ़ जा रही है. इधर भी सेंटर शुरू हो रही है, पेरिफेरी पर बढ़ जा रही है. लेकिन ये वाला स्टोन में क्या हो रहा है कि सेंटर पर ज्यादा है और पेरिफेरी पर कम है, जो कि इसका जस्ट अपोजिट है. उसी तरह से इसमें भी क्या है, सेंटर पर कम है और पेरिफेरी पर कम है और सेंटर पर ज्यादा है. तो इसका और इसका फॉर्मेशन एक तरह का है, इसका और इसका फॉर्मेशन एक तरह का है. तो कंक्लूजन में क्या निकाला हमने, मैग्नीशियम इज वन ऑफ द इंपॉर्टेंट मिनरल व्हिच इनहिबिट or allow further growth of gallstone depending upon the concentration change from the point of deformation of the gallstone, and this result is already published in atoms, and this result is supported by these two research. Therefore, the gallstone magnesium distribution from optical vision spectroscopy is clear and conclusion is a higher consumption of magnesium is associated with reduced risk of the gallstone. आगे बढ़िए. So again, अगर आप क्वेश्चन पढ़ते हैं कंसंट्रेशन ऑफ मिनरल में आपका मैग्नीशियम, पोटेशियम, कैल्शियम, सोडियम आ सकता है. Each magnesium, potassium, sodium intensity is higher at periphery as compared to the center, and if the आगे बढ़िए, and if the answer is yes, then you can get the gallstone like this, and if the answer is no, then you can get the gallstone like this. आगे बढ़िए. This paper is already published in compositional analysis with photographic spectroscopy. आप इसे लेके चलना. आगे बढ़िए. इन स्पेक्ट्रोस्कोपी का आपको एक्टा सम रिजल्ट पब्लिश. आगे बढ़िए. Now, अभी समय है डिकेसिंग का, कितना समय है? 5 मिनट है? नहीं है? 5 मिनट है ना? 5 मिनट में समराइज कर देता हूँ. वो तो गॉलब्लैडर स्टोन का था. अब किडनी स्टोन के बारे में थोड़ा बता दूं. Here you see, this is the kidney, and if the stones observed in this part is known as kidney. So these are the different types of the kidney stone. This is the your team. Here you see kidney stone again observed by another patient. Another patient, and this is one patient. This is one stone. And this is the intact one. I have got this. Again here you see this is the center part. This is the core part. If you see the then the magnesium at the periphery large. If the stone is so similar to that, multiple be 75. This is the absorbed part. Core part here in this case also smaller than at the center. In this is similar as the part by the similar way. Third layer, and third layer. We have the core part. Okay. So here also you see the potassium intensity. Sodium, this is sodium. Sodium is spectral line. It is smaller and this is great. Small. That like this one. So in Kamari, urinary bladder stone has shown bladder stones has similar trend as shown magnesium. Photograph of all my research is color. You see, Ashok Kumar Pratap, and he presented in this talk were done by my research intern Jaina Pajali, Ashok Kumar Pathak and Vivek. The other members Singh, he is the assistant professor in SPM degree. Kumar Pathak सिखा रहा है Kumar Tiwari and so forth, and the final DRDO is truly acknowledged. क्लियर देखिए कि आप एक पॉइंट पर पेज पर सेकंड पर किया, थर्ड पर किया, फोर्थ पर किया, तो लेखा ही दिखाई देता है। पॉइंट कर दिया, थर्ड पॉइंट कर दिया और देखा आपने और ड्राइंग हो गया था और एक गवर्नमेंट को देखा दूसरे पेशेंट से लिया गया था और वो इसमें बहुत सारे थे, दोनों का जो ग्रोथ है सिमिलर ग्रोथ तो सिमिलर नहीं होगा, हाँ, लेकिन सर एक बात है आप ये पूछता है इसका मतलब इसका अनसर्टेनिटी होता है। में खा रहा होता मनुष्यों में सब में नहीं होता है, ये तो यही होता है, बहुत दिन तक पता
This technique is consistent for only one laser art. It can be also done.
If you remember, in my surface should be greater than the continuous laser. It cannot achieve the laser irradiance at the sample surface, which is greater than 3.9 watt per centimeter. Because the power of the laser irradiance is energy per unit area per second.
सर मेरा ये क्वेश्चन था वायर लिकोग्राफी परफॉर्म करना है
नहीं ट्राई नहीं दिया
सर, अगर वायर लिकोग्राफी एक्सपेरिमेंट परफॉर्म करना है तो सर हमारा जो रेडिएशन होगा लेजर का वो 10 to the power -9 आपका वाट पर सेंटीमीटर
9 नहीं प्लस
उसके ऊपर का 10 to the power 9 पावर का।
हाँ।
सर अगर मैं अपना हेयर लेके अगर मैं उससे परफॉर्म करना चाहता
हाँ।
एक प्रोसेस ये भी है कि इतना ज्यादा रेडियंस होता है उसकी जो बर्न होने की प्रॉपर्टी होती है, इस चीज से हम कैसे कम आउट
ये ऐसा करते हैं कि जो लेजर का जो फोकस होता है बहुत ही स्मॉल एरिया में होता है। माइक्रो अगर उसका एरिया निकालेंगे तो माइक्रो सेंटीमीटर स्क्वायर से भी कम है, तो आप लेजर की प्राइस बहुत ज्यादा होती है तो स्मॉल एरिया से भी वो एब्रेशन होगा, पूरा पूरा लेजर जब पेनिट्रेट कर जाएगा तो बंद हो जाएगा। Any more question? Okay, next. If there is no any more question, so let's again thanks to our speaker. Thank you very much. हमारा पेन ड्राइव दे दीजिए सर, पेन ड्राइव ये
इसमें टारगेट
इसको कॉपी कर लीजिए, इसको कॉपी कर लीजिए सर, मैं आगे कॉपी सर का पेन ड्राइव दे दीजिए। सर वो जेब में पड़ा,
जिस लैपटॉप पे डायरेक्ट मैं खुद से आगे
ले ले अच्छा पेन ड्राइव दे दीजिए,
डायरेक्टर लेके चेंज कर दीजिए बस। एक ही प्रेजेंटेशन होगा एक बार चला के चल रहा है। इसको ऐसे कर दीजिए, मैं ठीक से उसको मूव कर दूँ। इसे डायरेक्ट
Okay. So after this fantastic talk of Professor A.K. Rai sir, we have another speaker, great speaker here, Dr. Sunil Kumar Singh from IIT BHU Varanasi. He is assistant professor in IIT BHU Varanasi. His area of interest is laser spectroscopy, photon conversion, and optical imaging, optical data storage, energy harvesting, etc., etc. So he has joined here in IIT BHU in 2013 actually as his past faculty as well as after that the assistant professor there. And he has recognized as a very reputed spectroscopist, physicist, and he published a lot of papers in this area of spectroscopy and laser spectroscopy too. He has a lot of fellowships. He got DST Inspire Faculty Award. He has postdoctoral fellowship, CSIR Nehru PDF, DS Kothari Fellowship, Senior Research Fellowship, listed as the world top 2% scientist. So he is in his own area. He is the top 2% scientist list of the Stanford University. And then ML Khurana Memorial Prize, Young Scientist Award, Indian Science Congress, Gold Medalist, first rank MSc in Physics from the same place, Best Paper Presentation Award, and a lot here in the listed here, a large number of here. And he is a member and professional, a large number of professional bodies too, member of Indian Science Congress, member of Laser and Spectroscopy Society of India. He has delivered several talks in national and international conferences as well as in abroad too. He has postdoctorate from abroad. Also, you can get much more information from the IIT site. After just typing Sunil Kumar Singh IIT, you can get the information. And he has a very good lab there in IIT Physics Department too. And several students, I think 7-8 students are still working as PhD students with him. So he is a very renowned physicist doing spectroscopy as well as a very good, very excellent teacher. And so he is going to deliver his talk here on the topic of laser spectroscopy for biological application. Laser spectroscopy for biological application is talk. So I invite Dr. Sunil Kumar Singh to deliver his talk.
Thank you, sir. Thank you for this very long introduction. First, I would like to thank the organizer, especially Bipin Bahadur sir. I was visiting Mumbai, VRC, and he called me to deliver a lecture. And he said that this lecture should be on lasers and its application in biomedical field. And then Dwivedi sir also talked to me and said that please try to confined in area of biological sciences. But when I saw the time for this lecture, it was around 45 minutes. So I cannot limit myself in biological application only. So the topic for today's presentation is laser spectroscopy for multifunctional application. But I am not a kind of very senior scientist. So my introduction should be like that. I am a student from UP College. I did my BSc from here. I did my MSc from here. And I proud to be a student of this college, right? Then I did my PhD from BHU, and so many things sir has already mentioned. And I will start with very simple, just like a fundamental teacher like presentation. So if I talk about laser spectroscopy, so what I am going to talk is, if we see photoluminescence, then if we have a system, when I say system means atom, molecule or solid, right? So here I have shown some levels, that means I am talking about atoms. So in case of photoluminescence, we excite a system, it goes to some excited state. From there it relaxes to intermediate state, and then it goes to ground state and gives some transition, particularly in visible and ultraviolet region, because optical spectroscopy is somehow related to visible and ultraviolet region. So this is called photoluminescence, traditional emission that we get in most of the cases. But what we do, where we need a laser specifically, is that if we have a system, we excite, it goes to some intermediate state, and it stays there for some time, and then it absorbs another photon here, and it goes to excited state, and from there it relaxes back to the ground state. What is the uniqueness in this case is, the emission has higher frequency than the excitation. So for an example, you can consider we are exciting by infrared light, and we are getting emission in visible and ultraviolet region. So this is a non-linear technique. You are exciting by low energy, and you are getting higher energy, which is completely different from photoluminescence. So you need specifically a laser. You can do such kind of spectroscopy only by exciting your sample by laser. So now I can say that I am going to present some work which needs laser. Okay. Now this kind of non-linear spectroscopy is already known in terms of simple harmonic generation, that you know in case of Nd:YAG laser. In Nd:YAG laser, we have emission at 1064 nanometer, and then we have frequency doubling 532, 355, and 266 nanometer. MSc students know about this laser very well, Nd:YAG laser. And in case of two photon absorption, this is also a kind of process in which you excite by low photon and you get high photon. But in these two cases, you see there is the involvement of virtual states, and virtual states have a lifetime of the order of femtosecond. So this kind of technique can be done by laser, but the laser should be high density laser, 10 to the power 6 to 10 to the power 9 watt per centimeter square, and you can do it by continuous wave laser. So somewhere 10 to the power 6 watt is a very high laser you need to do this kind of spectroscopy technique. One more technique is there for this kind of non-linear spectroscopy, that is called triplet annihilation, and that usually takes place in case of molecules, because this triplet state is possible only in molecules. In atomic state, there are electronic state, and in triplet, this is the contribution of electronic, vibrational, and rotational state. All is there. So by combining two triplet states, you get a emission which is having higher frequency than the excitation one. So two triplet state by cooperation gives an emission which is in higher frequency to that of excitation. But in all these three cases, you need a laser which should have a high density flux for excitation. But in case of this photon upconversion spectroscopy, all the levels involved are real level, so that's why you can do photon upconversion spectroscopy, but with a very low power laser. I can say in milliwatts. You need to have a diode laser, very simple diode laser, a few milliwatts, and you can do photon upconversion spectroscopy. Okay. As I said that in this case, we need a system in which there are real levels, intermediate spectroscopy. So that in case of lanthanides, we have F to F transition. When we call about F to F transition, that means these F levels are shielded by 5s and 5p levels. So this is a transition which is shielded by some orbitals. So the effect of outer environment is very less. Okay, so next I am going to say that we use nanostructures to get up conversion. So why we need nanostructure when I am saying that there is no effect of environment? So only nanostructure is used because of the application purpose. Okay, there are many ways how we can get up conversion, but today I will talk about a kind of up conversion in which YB is used, and in case of YB there are two states, ground state and excited state. So you can say two YB ions cooperatively combine two photons and then transfer their energy to some activator ion, and by that way you can get the photon up conversion based emission which is higher in frequency to that of excitation. Like this is the difference between these two levels is around 980 nanometer. So by using YB and an activator say erbium, one can get emission in green, red and blue region. Okay, how we can do it? In case of photoluminescence, we know that we need a source to excite our sample, and then we use a monochromator and send the signal to detector. But in this case, we need only a laser. It will excite your sample, and the emission will be detected by a monochromator having a PMT as a detector. Okay, so I am coming to a real system where we can see the upconversion emission. In this case, there is a host, gadolinium oxide, which has been doped with erbium and YB. As I said, YB will be used as a sensitizer, and erbium will give you upconversion emission. We are exciting by 976 nanometer laser, and we are getting emission. You can see so many transitions, and in visualization, you can see we have three colors: red, green, and blue. We are exciting by infrared light, and we are getting emission in visible and ultraviolet in blue region. So it is very clear that emission is having higher frequency to that of your excitation. Okay. Now directly coming to the use of this kind of spectroscopic technique in biological field. Okay. In case of bioimaging is a technique by which you can see the structure inside your body, like in tissues or in tumors, cancers. There you can visualize the real structure there. So the technique that is used traditionally is there is a fluorescent level which has been tagged with your biosystem, like you have a tissue and you have tagged it with some organic dye, and then you are exciting with UV light. But when you are exciting with UV light, along with your dye, there are some organic pigments inside the body which are excited by UV light. They themselves create the emission, and this emission creates interference. You are getting one emission due to your sample. At the same time, you are getting one emission from your specimen itself. So your signal noise ratio is very low. It should be very high. So in order to avoid this, upconversion material, and we can excite with 980 nanometer. This 980 second thing is that the penetration autofluorescence from your specimen itself, and at the same time penetration depth will be high. So you will have high spatial resolution with penetration depth, right? Okay. So what we did, we synthesized a system of sodium scandium fluoride. Why fluoride? Because fluoride can be synthesized in a liquid form. If you are targeting any biological application, then you must have your sample in liquid form. Then only you can inject it inside the body. So we synthesized sodium scandium fluoride. Activator was same, erbium and YB. So YB is absorbing 980 nanometer, it is transferring this energy to erbium, and now your erbium can give emission, intense emission in red and green region, and the blue emission is very weak. So what I will say that you will excite your system with 980 nanometer laser, and you will get emission in green and red region, right? So platform is ready. We synthesize the particle size, and particle size was somehow 20 nanometer only. So if you have a small, very small particle size, then you can make a suspension of this kind of material in liquid form, and that is very stable. We have monitored its stability up to 2 weeks. There is absolutely no problem. So it is a fantastic material to do bioimaging. Now what we did, we take HeLa cells and we injected this material in HeLa cells and visualized the internal structure of cells in green and red region. So what I can conclude here is that photon upconversion spectroscopy, which is done by nanomaterials by excitation of laser, can be used for bioimaging of different kind of cancerous cells. This is some advanced work that are going in collaboration with zoology department, because I am not expert. What they are doing is they are encapsulating this upconversion material with some targeting molecules. These targeting molecules are specific to attach with a specific cancer. So these two molecules are there, polyethylene glycol bilirubin and a long name, some complex. These are two molecules, you can understand. These are two molecules which can specifically hit the triple negative breast cancer cells. So if you have breast cancer, then this kind of upconversion material coated with this molecule will directly attach to the breast cancer cell. So this work is in progress. Okay, the second part of this talk is somehow related to the climate change. So climate change is like solar cells are used to harvest the energy. So our idea was, I will present in a very simple way that a MSc student can understand. You see this green curve. This green curve is for the conversion efficiency of silicon solar cell. You have many solar cell maybe in this institute. So the best response is in this region, from 500 to somewhere around this region. So best response you can see, and the response in this UV region and infrared region you can see is very poor. So our idea is that if we can make a material which can absorb in UV region and can absorb in infrared region and can emit somewhere here in green and red region, then that material could be used to improve, not act as a solar cell element, but it can improve a already existing solar cell, and it can improve its conversion efficiency. So what we did, first type of material, you know that you can use an upconversion based material. You can excite with infrared light somewhere here, and then it will emit in green and red region. The second material that we selected, which can be excited by UV light, conversion efficiency is poor somewhere here, was an organic complex which was having europium 3+ as an activator. So this can be excited by UV light, and it can give emission in 611 nanometer, means red region. So this two material gives emission in green and red region. So behind this work, there were three basic questions. If you want to do anything, then there are some basic questions. If you are going to mix two material, so the first question is what type of interaction was there? By what interaction they were bound? This was the first question. And the second question, if there are two optical elements and they are bound together, so is there any new optical effect, or they are individually behaving separately, right? And the third question was, if you have prepared this material, idea is very fantastic, but if you test it on the real ground, is it going to work as a energy harvesting material or no? So keeping these 3 points in mind, we did this work. It is a old work we did in 2017, but to meet the objective of this conference, I am selecting this talk for today's presentation. So I will not go into deep detail of this, how you are getting emission by upconversion, because you already know. And then For the second material, what I would like to say that there are two organic molecules, FEN and TTA. When we excite this material by UV light, first FEN is excited, it transfers its energy to TTA, and this TTA finally transfers its energy to europium 3+, and then you get it's a deep spectroscopy, molecular spectroscopy. First material gives emission by atomic spectroscopy, and the second material is giving emission by molecular spectroscopy, right? So you can say that what we are getting is something like cascade kind of energy transfer. These two materials are behaving like antenna to harvest the solar energy, and finally this europium gives emission in red region, because our target is to get emission in red region by absorbing the UV light. Okay. This is the microstructure of this hybrid material. What I can conclude is that the particle inside this hybrid is of the order of less than 10 nanometer. Here you can see this is the TEM picture, TEM micrograph of your upconversion nanoparticle. Your upconversion nanoparticle is a nanocrystal, and organic material is just an organic molecule, I think. So the size of your upconversion nanoparticle is below 10 nanometer. This is very important for this material. Second, these upconverted nanoparticles are very uniformly dispersed. I will not say that they are bound, but they are dispersed in the organic material. So they make a teeth-like structure. And this upconverted nanoparticle retains its crystalline nature even in hybrid nanoparticle when we have both this material. Okay. We perform type of interaction study. Then we recorded the X-ray diffraction of both the material, means upconversion nanoparticle and then hybrid nanostructure. And you can see that all the peaks are almost overlapping. That means there is no any crystal related change in the hybrid nanoparticle. Upconversion nanoparticle is having its own crystal structure even in hybrid nanostructure. Then we recorded the Fourier transform infrared spectroscopy, and I can consider that you know about this technique. This technique tells about the functional group present inside a system, and functional group is a signature of molecules present in your system. So if you compare, the first one is upconversion nanoparticle. There is no obvious FTIR peak there. These are the peaks which are present in every FTIR spectroscopy, because these are due to carbon dioxide and some water molecules. But these two are interesting. You see this organic material and then hybrid nanomaterial. So we matched all the peaks, and we found that almost all the peaks are matching. So there is no change at the molecular level also. So what we can conclude there, that there is a kind of van der Waals type of interaction. There is a very weak interaction. You may ask question that how you can say that there is an interaction, because here we are not able to say anything positively, but what conclusion is negative, that there is no change in atomic structure, there is no change in molecular structure. So how I can say that there is some interaction? That you can prove by photoluminescence. So in the next slide, you see this is the absorption spectrum. You can see these are the absorption peaks due to organic molecule, and this is due to lanthanide present in your medium. You can say that your material can absorb in infrared region, it can absorb in UV region. That's fantastic. And you regarded the emission of europium 3+ in your organic molecule. It gives characteristic bands due to europium 3+. That is also okay, nothing new. But when we excite these hybrid nanoparticles by 980 nanometer later, we get all the peaks that are due to europium 3+. That is also okay. What is new? New is this peak. You see the energy level structure of your organic molecule. If you are exciting by 980 nanometer later, your excitation is somewhere here. There is no peak in organic molecule due to FEN, TTA or europium that can absorb 980 nanometer. But when you are exciting by 980 nanometer, you are getting emission due to europium 3+. That is a new signature. And it is possible only if then two systems are interacting, and a spectroscopist can say that there is some kind of energy transfer from one system to another system. I am not going to very deep and I will say you how energy transfer takes place. Here you see, when you excite your upconversion nanoparticle, then there are two levels, quadrant S 3 by 2 and doublet H 11 by 2, which are responsible for the green emission in erbium 3. And these are in almost overlap with 5D0 level of European 3. So what we propose that there is an energy term when we are exciting by 980 nanometer laser. This green emission of erbium is being transferred to European 3+. Mind that it is not radiative energy transfer process. This is non-radiative energy transfer process. Non-radiative energy transfer takes place by collision. It's a kind of collisional energy transfer. It is not like that it is emitting a photon and that is being absorbed by European 3+. Rather, they are very close enough. What I am saying that they are making a hybrid. When they are making a hybrid, then they are very close enough. And when they are very close enough, then there is a non-radiative energy transfer from one system to another system. So by this way, when you are exciting by 980 nanometer, that cannot absorb by the organic molecule, even you get the emission of organic molecule when you are exciting your inorganic system in the hybrid. So by this way, what I can say is there is some interaction. Since there is no any interaction at atomic and molecular level, so the only conclusion is that there is some weak interaction, which is van der Waals type of interaction. And for an effective van der Waals type of interaction, you must have a high surface area, and that you get by small nanoparticle, which is in our case less than 10 nanometer. So we are now clear with the two questions. This is the deep spectroscopy by which you can prove the energy transfer process. So if erbium is giving its energy to European 3+, so in principle the lifetime of erbium 3+ would go down and the lifetime of European 3+ would go up, and that we get. So by decay curve we also prove this. The last part that I want to convey is, the last question was, can this material be used for getting energy harvest? So what we did, we dispersed this hybrid nanoparticle in some polymeric substrate, and this polymer was polymethamethyl acrylate. So what we are doing, we are making a thin film which is loaded with our material, and the loading polymer is PMMA, which is transparent. So your PMMA is not absorbing anything. Ideally, I can say there is some absorption, right? Around 3 to 5% absorption is there, but 97% it is transparent in the region of interest, and your material will do all the things. Okay. Now, in order to see the conversion efficiency or to characterize any photovoltaic cell, there are three parameters that we have to see, and these three parameters are ISC, short circuit current, off circuit voltage, and fill factor. So what we did, we coated this film onto the surface of a crystalline silicon solar cell, and then we excited with different wavelengths and recorded these three parameters. We found that when our material is dispersed in polymer medium with 2.5 weight percent, जितना weight percent PMMA का है उसके 2.5%. When it is dispersed in a quantity of 2.5 weight percent, then there is a signature which is positive, and it tells that it could enhance the conversion efficiency of a pristine crystalline silicon solar cell. You can see RV parameter showing some significant increase. But if you are increasing the concentration of your material dispersed in the polymer cell, so the increase is not significant. So I can say that you have to optimize the concentration which will be dispersed in polymer, and in our case it is around 2.5 weight percent. So we can conclude, okay, I will not go in third one. So what I can conclude is that there are certain lanthanide based material that can be excited by laser, near infrared laser, and it gives emission in visible region, mainly in green and red region, and I try to convince you that this kind of material can be used for bioimaging purpose, which is directly related to biological or biomedicine purpose. And secondly, I try to convince you that by making a hybrid of this upconversion nanoparticle and a simple fluorescing material, we can make a hybrid nanoparticle which can be dispersed in thin film form, and it can get on the surface of crystalline silicon solar cell, and in this way it can improve the conversion efficiency of silicon solar. So for today's talk, there was a third part also, but I can understand that it is around 3 o'clock, so we are being very late for the lunch. So I will stop my talk here and will finally reach to. This was a kind of collaborative one. You can see that there are many aspects which we have to address. I cannot singly do it. So these are the collaborators from my institute and outside with whom I collaborate to do this kind of transdisciplinary research. I have 4 PhD students, direct PhD students to me. There are 3 students to whom I am serving as a co-PI. 1 postdoc in my group, and I have funding from Department of Science and Technology, New Delhi, and also from my institute. All the facilities are being given by my institute, so I will acknowledge it. And lastly, I will thank you.
Okay, so the talk is now open for question. Any questions from the audience?
हाँ बताइए.
क्वेश्चन ये था कि अगर मान लीजिए सपोज दैट सर एक हम वेव फंक्शन, सपोज दैट सर साई एक वेव फंक्शन, ठीक है, दूसरा नंबर
नहीं पूछो ले.
सपोज दैट मान लीजिए सर हमारा सन है, उसके ऊपर से बाहर से हमने कोई सर लेजर ऊपर फॉल करा है, ठीक है, उसे अगर हम थोड़ा वेव फंक्शन में जाते हैं, ठीक है, तो ये साई अगर मान लीजिए सर ये साई को भी साई आता है, ठीक है, नॉर्मली सर, ठीक है, ये दोनों जो साई का नेचर वो सिमिट्री होगा. इट मींस कहने का मतलब ये है कि हमने अर्थ पर सर एक बाहर से लेजर को उस पर फॉल करवाया, ठीक है. अब क्या है, वो सन का नेचर तो चेंज होगा, और चेंज होगा तो फिर वो पहले वाले सन के कंपेयर में सिमेट्रिक रहेगा कि अनसिमेट्रिक होगा.
Okay. So it is a kind of electromagnetic to electromagnetic light interaction. Like when you are saying sun, it is not material. So it is not light matter interaction. It is light to light interaction. So in this case, these two wave functions will interact, and in this case, you will have a combination of both the wave functions. So if it is psi1 and psi2, then you have the output which will have the information of psi1 and psi2. I can say A psi1 plus B psi2. That's what I can say, right?
सर क्या है सिमेट्रिक नेचर इशू कर सकते हैं अगर दोनों होंगे, कुछ सिमेट्रिक एसिमेट्रिक, कुछ हालांकि सर देखा जाए तो सर ये अनसिमेट्रिक होना चाहिए.
हाँ,
तो उसके बारे में कुछ कहना चाहेंगे सर, बता दीजिए.
जब तक आप पार्टिकुलर नहीं होंगे, सिस्टम को डिफाइन नहीं करेंगे, तब तक आप सिमेट्री डिफाइन नहीं कर सकते. इलेक्ट्रोमैग्नेटिक वेव्स का नेचर जब तक नहीं करेंगे, दोनों वेव फंक्शन डिफाइन करके ही तब पेरिटी अप्लाई करके आप वहां पे आप सिमेट्रिक और एंटी सिमेट्रिक वेव फंक्शन डिफाइन करें. ऐसे जनरल केस में आप सिमेट्रिक और एंटी सिमेट्रिक वेव फंक्शन की बात नहीं कर सकते. इट्स अ काइंड ऑफ you want to know my own opinion about this, but I can say only if I know both the electromagnetic waves clearly, because wave function is always dependent with boundary conditions. So here you are not defining the boundary condition, you want some answer from me. It is not possible, right?
Any more questions? Okay, if there is no any questions, then let's again thanks to the speaker.
इसके बाद हम लोग लंच के लिए चलेंगे फिजिक्स पार्ट में, और लंच के बाद आधे घंटे हम लोग आएंगे लेक्चर और लंच के लिए फिजिक्स पार्ट में चलते हैं. लेट हो गए फिर आधे घंटे में करने कोशिश करेंगे. फिर भरने पर हम लोग यहाँ फिर वापस आए.
इसमें कोई कॉपी नहीं की तो डाल लेते लैपटॉप क्या फाइल का नाम क्या है?
दो बच्चे हैं
भैया जो हमने कराया है तो इसकी स्पेलिंग चेक कर लीजिएगा.
यही वाला
प्रेजेंट करके देख लो। लेकिन
सर थोड़ा इसमें देख लेते हैं, कुछ प्रॉब्लम आ रहा है।
प्रॉब्लम आ रहा है क्या?
नहीं सर ठीक है।
सर अब ले लेंगे अभी ये